Welcome to topic 2 of our IB biology course. Now we are looking at molecular biology. This is the first video of topic 2 and we're looking at molecules to the metabolism. Okay, so when we're looking at molecular biology, what we're looking at is all the chemical reactions and processes that take place to allow life to occur. Metabolism are all those chemical reactions that are occurring within the cells of living organisms. It's a relatively young science. We're only looking at 50 to 60 years old. And I guess the question is, why would that be? And that's with the discoveries of microscopes and the research into biochemistry and those kind of areas have opened up this whole area of biology. Okay, so molecules to metabolism, two really key words just in that title. The first one being molecules. So what are molecules? And they're this incredibly diverse group of atoms joined together to make a molecule, okay? So we have individual atoms and then more than one atom joined together, whether they're the same element or not, makes us that molecule. Now, in biology, we have a real focus on these carbon-based molecules that make up living things. They're an incredibly diverse group, including our carbohydrates, lipids, proteins, and these nucleic acids. Alrighty, so that's where we're going and heading in terms of molecules. Now, what about metabolism? Okay, so metabolism, this is saying that web of all the enzymes catalyzing reactions within cells, okay? Metabolism are those reactions occurring in the cells, and they would not be occurring if it was not for the enzymes to catalyze them. And we will look further into that as we move through this topic. So a couple of few new words. We've got this anabolism, okay, and this is the synthesis of complex molecules. Catabolism is the breakdown. So synthesis is building and then breakdown, so they're our opposites, okay? Anabolism building up, catabolism breaking down. Alrighty, so one of these key points of your IB course is understanding the falsifications of theories. So there are theories that existed and were well believed until science has allowed their falsification. Okay, and we've got one of those in this topic. The artificial synthesis of urea has helped to falsify this theory of vitalism. So let's have a look at what vitalism is. Okay, so vitalism, this was the belief that living organisms were fundamentally different from non-living entities in their particles. Okay, so living entities contain a non-physical element that makes them unique. Compounds that are produced by living organisms, such as sweat, blood, urea, etc., require this non-physical vital element in order to be made. The vital element is sometimes equated as the soul. Therefore, these products cannot be made outside of a living organism because that vital element needed to be present. And a rock, for example, doesn't have that vital element. A rock doesn't have soul. So it was believed that that vital element that was non-physical needed to be present for life to occur. Okay, so let's think about urea. It's a naturally occurring nitrogen containing compound and it's made by living things. It's relatively simple in structure. You can see its structure there. It's a component of urine. Okay. Excess amino acids in the body equals excess nitrogen, which has got to be removed. Okay, so in our metabolism, we're breaking down proteins. We are needing to remove excessive amino acids. Okay, so somehow key component of amino acids is nitrogen. Somehow we need to remove that nitrogen. We will look further into amino acids, protein structure, and things like that as we move through this unit. But just remember, amino acids contain a lot of nitrogen, and when they're broken down, we need to remove that somehow. Now, a series of reactions occur in the liver to convert the amino acids to urea to be taken via the blood to the kidneys, and then this urea becomes excreted from the body as urine. Okay, so here we've got our chemical formula for urea. So we can see we've got that carbon and oxygen, and we've got nitrogen and hydrogen in there. So you've got your molecular structure and your chemical formula there. And there is its molecular structure via molymod. Okay, so this falsification of the vitalism. In 1828, Frederick Waller synthesized urea using silver isocyanate and ammonium chloride. This was the first organic compound to be synthesized artificially, so it was a major step in biochem. This discovery enabled scientists to determine that urea can be artificially synthesized using this equation. So if you've got ammonia plus carbon dioxide, your products are then ammonium carbonate, urea, and water. This is a different reaction to the one that occurs in living organisms, but it is showing that urea can be synthesized. We now accept that processes in living organisms are governed by the same chemical and physical forces as in non-living matter. So basically, there is a reaction occurring in living organisms that produces urea, and it can also occur outside of living organisms. However, some organic compounds have not been synthesized artificially. For example, hemoglobin, we still haven't figured out a way that this can be synthesized outside of living organisms. Alrighty, so as we move through chapter 2, and as I've already said, we are going to look closer at many of these molecules. Okay, so we're going to look closer at the importance of water in metabolism, carbohydrates, lipids, nucleic acids, and proteins. So they are all to come in future videos. But before we get there, let's just revise some of these things that we probably already know, and if we don't, it's a good time to have a think about them. So first of all, our carbon compounds. Carbon has the ability to form 4 covalent bonds. Okay, you should remember that from your earlier years of science. However, if this is a bit of revision and it's a bit rusty, don't panic. This leads to massive diversity in the compounds that it can form, because it's got 4 choices of elements that can bond, or molecules that can bond to it. Now, carbon can make rings or chains of any length. For example, your fatty acids can be up to 20 carbons long. Okay, so we've got an example of a ring molecule there, and that's a chain molecule there. So these are our carbon compounds because they contain carbon. Okay, so some examples of our carbon compounds here. We've got our carbohydrates and our subcomponents. So they are the basic building block as such of each of these. Don't use that term basic building block. I'm just using that to imprint the message into your mind. So carbohydrates, we're going to say subcomponents would be our monosaccharides. So one sugar, monosaccharide, carbohydrate. Our lipids, and those subcomponents are our glycerol, fatty acids, and our phosphate groups. Our proteins, our subcomponents are amino acids. So polypeptide, peptide, being the bond between the amino acids, and then our nucleic acids, the subcomponent is the nucleotide. Okay, so we focus on classifying carbon compounds as carbohydrates, lipids, proteins, or nucleic acids. These are the four main classes of carbon compounds that make up living things. They're all different properties. They all have, different properties that are suited to their function. And we'll cover these four compounds in greater detail as we move through the topic. As I said, we're going to focus on our carbohydrates, our lipids, our proteins, and our nucleic acids. And I don't know that they all arrived as they should have done there, but we've got that table showing those sub-blocks. And if we look over here, we've got our nucleic acids building up our DNA there. We've got our amino acids here building up our proteins. We've got our fatty acids here, and we've got our carbohydrates here. So each of these small shapes is a monosaccharide there. Okay, so classifying our carbon compounds. Okay, so as I said, several times now, we've got those carbohydrates, proteins, lipids, nucleic acids, and within each of those we have our subcategories. So if you look at our carbohydrates, you see we've got monosaccharides, meaning one saccharide, that saccharide being a type of sugar, and then we've got four different types of sugar there, so glucose, galactose, fructose, and ribose. Disaccharide means two sugars joined together. So two monosaccharides joined together gives us our disaccharide. So our maltose has two molecules of saccharides joined together, lactose, sucrose. And then polysaccharides mean many sugar molecules joined together. Our proteins, okay, and there's some examples of our proteins. Our lipids, again, we've got triglycerides, would suggest that we've got three glycerides there. We've got our phospholipids and our steroids, and then we've got our nucleic acids, okay, and we've got examples of each. And we're going to look further, as I've said, into each of these in their individual topics to come. So a few bits of revision that could help you in our unit that we're about to do. Okay, monomers, single molecules. So if we're looking at our carbohydrates, our monomer is our monosaccharide, one unit. Polymerization causes a polymer to form, so that's a long chain, many units. As we've already looked at, our organic molecules, we've got our four categories, our monomers, and then the names of our polymers. That is the end of 2.1. I'm looking forward to seeing you in 2.2 when we can further investigate these biological molecules.